In this video we will discuss about how the glycans can mediate the immune response. So whenever the pathogen or any uh, foreign harmful substance impact or try to attack the host cells, its immune response will get activated and act against those harmful foreign substances. So how this glycan plays a role towards this preventing the attack from the foreign antibodies, a brief description is shown here in this figure. So in the first figure, that is on the left side, it has shown that the glycosylated MUC1 and this glycolipid that has galactans can interact with these glycan structures, that is P glycan structures that are present on UPEC, or this glycan structure that are presented on MUC1 can inhibit the step adherence. So, and these are known as direct interactions, and also the silated glycans can interact with this flu that is present within the hemagglutinin. So hemagglutinin can interacted with this silated glycans. So these are the three different ways by which host pathogen interactions can occur, and these interactions are mediated by the glycans. Now, how the immune system gets activated by recognition of this foreign harmful substance is shown in this middle figure, which is headed as this microbial recognition mediated glycans. So in this case, when the foreign antibodies are detected, when the foreign not antibodies, but when the foreign harmful substance are detected by this toll-like receptor 2 that are ubiquitously expressed on the host cell surface, so it can detect this PSA or PAM CYSK, uh, and thereby it will trigger this NF kappa beta uh, signaling pathways uh, by through and this tolerate through this tolerate receptor 2 activations, and it can generate more the toll-like 2 receptor glycans modulate and helps in this binding of this CYSK, and thereafter, after identifying the foreign antibodies, it can show this foreign, it can transfer this information about this no, not antibodies, foreign harmful substance epitopes information will be transferred to a major histocompatibility complex 2, and that transfer is also done by this N complex glycans, and thereafter this what will happen is this major histocompatibility complex will help in diverting this foreign harmful substance towards the endosome or lysosomes. That is shown here in this figure 2, and in the third figure it is shown in slightly in more in depth about how the B cells and T cells get activated. So the CD22 or we can say that SIGLEC2 can interact with the silated proteins antigens, and thereafter it can and these are expressed on the B cell surface, and it's known as B cell receptors. Thereby, when the silated protein antigen interact with the CD22, it can trigger apoptosis, and the acylated protein antigens will trigger the proliferation of the B cells. So in this case, and in this example, it is shown here that this silated protein antigens are the self antigen, whereas acylated protein antigens are the non-self antigens. Thereby, it will trigger proliferation of the B cells, and B cells can then function against those foreign harmful uh, substances that are in the host, that are trying to attack the host, or that have already attacked the host. And here the immune interactions are shown in the form of antibodies, where we can see that silated antibodies will trigger anti-inflammatory response, or we can say it will try to suppress the immune response, whereas non-silated antibodies will lead to the pro-inflammatory response, so it will upregulate the immune response. So in this example also, the silated antibodies are generated based on the silated antibodies are you can say, are the part of the host, and it will not generate immune response, whereas non-selected antibodies are the antibodies that are generated against the foreign harmful substance, and that needs to be taken care of by increasing the by generating pro-inflammatory response. And in the case of T cells, what will happen is that T cells uh, Galectin-3 ratings will increase the T cell receptor activations threshold. So what does this mean is that this galectin-3, when interacted with these T cell receptors through these glycans, it will increase the threshold. It means that the amount of foreign harmful substance that are required to activate the T cells will be increased, and these galectin-3 and T cell receptors are interacted to each other. And these interactions usually occurs when the amount of foreign harmful substance are low within the host cells, within the host body. So in this case, how the immune system gets modulated by the glycans. If you need to understand in detail how the glycans uh, in depth, how the glycans step by step mediated or plays a role in the immune response, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.